how you might develop tolerance, dependence, withdrawal from each substance. But there's going to be at least bits and pieces where you're going to see some research and some data that's been presented in animal data, as well as some human data of how we think that addiction develops, how tolerance develops, these kinds of things. All of the studies that are here, you should have enough information in the slide deck that if you really want to go read more about that you can. But we'll hit the high points of at least the neurobiology of addiction. It again also we could spend hours and hours and hours on this particular topic. So the main things is we're going to look at the anatomical pathways that are involved in the reward center. We're going to look at the neurotransmitter systems briefly. We'll look at dependence, tolerance, withdrawal, and then we'll hit briefly on some of the medication treatment side, some of the things that are available for medication-assisted treatment. So what is addiction? I don't have to read to you guys so you can actually see this, but it's basically where you're either ingesting a substance or you're engaging in some sort of activity that provides some sort of reinforcement or pleasure, but then by doing it, you start to interfere with your personal responsibilities, so work, relationship, health, those kind of things. So it's not a problem if you want to go to the racetrack and bet on the horses every once in a while, unless you decide that you're going to start skipping work and you're going to start maxing your credit card out to go and bet on the ponies, all of the time. It's not an issue of addiction if you periodically want to go have a drink at the local dive bar, but it is if you now start having to drink first thing in the morning when you get up, or you're finding that you're binging every weekend and it's starting to affect your relationships and so on and so forth. So I told you I was going to stay non-political because we were voting on 788, but I'm going to bring something up now that it's passed. One very good argument that I've heard, and actually Dr. McNeil and I had this brief conversation, that so alcohol is an addicting substance if used improperly, but it's legal. And you can go to the bar and have a drink, and if you have a single drink, you can leave the bar and most likely not be impaired, correct? Is there any possibility that you can utilize THC and consider yourself non-impaired? I don't think so, but that's just my personal opinion, just something to think about. Okay, so the cycle of addiction is not just neurobiological, it's just not neurochemical. It's not just genetics, it's not just environment, it's the collection of all of these things interacting with one another all the time. So we have things within us, both from a psychological standpoint as well as a genetic standpoint, that may be Pandora's box that can set us up for addiction or addictive type behaviors. So we know that our genetics can play a role in that. We know that our social situations can play in that. You all obviously have had some interaction with folks who have dealt with substance use or alcohol use disorders, and somebody had mentioned briefly looking through the patient chart and saw that they had some sort of traumatic event. So we have things that happen to each of us that can be triggers. We also have things within us because that have been passed down by our family lineage in our genetics that can also trigger the potential for misuse or abuse of substances. And actually the Health Sciences Center has some very prominent alcohol researchers, and they've done a world of work through the program that I got my master's degree through, basically the behavioral neuroscience side, and they've done a lot of alcohol research and looking at genetics and alcoholism, and so it's a real deal. So you've had neuroanatomy, so none of these terms are going to be unfamiliar. You have some very specific neuroanatomical areas that are either responsible for the production or release of neurotransmitters, or that are responsible for the binding of those, that have a direct relationship to reward pathways, memory, encoding memory and emotion, that all have to do with the development of substance use or substance abuse related disorders. I doubt we would have someone that would initially utilize a substance and continue to utilize it unless they got initially some sort of reward from that. Now, later on in the process of dependence, we start seeing individuals that are using because of the negative side effects of withdrawal, and they're trying to thwart withdrawal, but in the beginning, it's pleasure that we're attempting to gain from whatever it is we're engaging in, whether it be some sort of activity or whether it be some sort of ingestion, and we're pairing emotional latent events with that pleasure, and that involves multiple areas of the brain. And so we have several different areas involved, and you've been through each of those, so I'm not going to belabor what is locus coeruleus, what is the ventral tegmental area, what is the amygdala, where are these things at in the brain. You've been through those in neuroanatomy, correct? Okay, you passed Dan's exam. How did we do on the last exam? Everybody did good?
Everybody passed straight up from the course.
Good. Nobody got an ass chewing because they weren't here whenever I lectured last time when we took roll?
No.
No? Okay. Just making sure. Dan just is getting kind of soft, and Dr. McNeil getting softer as he gets older. Okay, so most important pathway in reward is going to be dopamine. Dopamine, 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 dopamine. Okay? So dopamine pathways, reward, pleasure, euphoric sensation, but also dopamine has to do with motor function. So you may have talked about Parkinson's or Parkinsonian things, so dopamine is directly involved in that. Dopamine is also directly involved in a number of psychiatric conditions. So you think of schizophrenia specifically, you think of other mood disorders, major depressive disorder and so forth. You have dopamine that's directly involved in mood. And dopamine is directly involved in decision making. So your orbital frontal, prefrontal cortex, your start-stop behavior, your yes-no, it's sort of the green light, red light, yellow light of your brain is in that frontal cortex, orbital frontal, prefrontal areas that all have dopamine connections. So you're sending a number of neuronal projections that all have relationships to dopamine from ventral tegmental area and nucleus accumbens to hippocampus, amygdala, prefrontal cortex. And so the hippocampus is involved because of memory, the amygdala is involved tying emotion to memory, and your prefrontal cortex involved in starting, stopping behavior. We're going to see later on in the lecture how important this prefrontal cortex area is and why substance use in younger individuals, especially adolescents, is so important as to why they can develop addiction and addiction tendencies, because the frontal areas of their brain are not fully developed when they start looking at utilizing these substances. So just from a rudimentary perspective, the back part of the brain develops much faster than the front part of the brain, okay? In women or young ladies or girls, earlier than boys. That's why boys are stupid longer. They do dumber things longer. They tend to engage in more reckless behaviors as they are younger because this orbital frontal prefrontal cortex is immature. And so the ability to start, stop, yes, no, good, bad is not fully developed there. It's not an excuse, but it's just kind of how boys are, and they're all stupid. Serotonin, that's another big one. So we know that serotonin and dopamine are involved in reward pathways, so it would make sense why serotonin and dopamine are also involved in mood disorders, correct? And one thing we're not going to, because there's so much involved just with dopamine, we're not really going to get to go through norepinephrine. So we know that norepinephrine also has a large relationship to reward pathways, less so than dopamine and serotonin, but that would make sense why norepinephrine has such a large relationship to mood and mood-related disorders as well. So the big thing with serotonin pathways, unlike dopamine pathways from the ventral tegmental area, serotonergic pathways originate from where? Where is this thing? Where's the raphe nuclei located? Collection of nuclei in the, sort of in the brainstem, right? It's a little further back in the old brain, okay? So it's involved in mood, memory, sleep, cognition. Why do you think that these reward pathways and these mood and memory pathways, cognition pathways, why do you think that they're in more primitive areas of the brain? It's how we function, it's how we survive, right? So it's how we, if you're the squirrel looking for nuts, you A, want to remember where the nuts are, and B, you want to remember where the good tasting nuts are versus the bad tasting nuts. And you want to remember where you hid the nuts that you're going to use to eat through the winter. And it's all about species preservation. And so a lot of these pathways are in older or in more primitive areas of the brain and are ones that develop earlier in our developmental periods, okay? When we're born, we have to be able to engage in certain physiological functions to survive. We've got to breathe, our heart's got to beat, we've got to digest, these kind of things. So a lot of that control is done in more primitive areas of the brain, okay? When you're an infant, you're not thinking about where do I go get food and how do I get food and where do I remember where that food is and those kind of things. You're relying on somebody else to teach those things or provide those things over time as that portion of the brain develops. And isn't it interesting that we as humans are the most, supposed to be, the most advanced beings on this planet, yet the more advanced the being is, the less we're able to do for ourselves at birth as compared to other animals that are born into life onto this earth. Because you think about what, I mean, the baby giraffe, dude, you got to get up and run here pretty quick, or you lay there too long and the hyena ate your dinner. So you've got to think about why is that? And are their brains different than ours? Absolutely. They don't have a large cortex, so they're not doing a lot of the things that we're doing, but the primitive areas of the brain are having to be up and running very quickly. And that has to do a lot of with these dopamine and serotonergic pathways, and that's why we know so much from animal research about the neurobiology of some of these conditions because of utilizing animal brains to study those. So reward pathway is not just a theoretical Pandora's box. It's truly a set of axonal networks that's activated by a large number of things. This could be whatever provides you reinforcement or pleasure. It could be food. It could simply be the Dr. Pepper that I love to drink all of the time. It could be cigarettes. It could be cocaine. It could be alcohol. It could be marijuana. It could be whatever. Roller coasters. Who likes roller coasters? Who's ever been to Cedar Point, Ohio? Sweet, I was there last week. Yes.
My dad's family's from there. So
Okay. I only rode 10 of the 18. Cedar Point is the tied in the world for the largest collection of roller coasters in the world. There are 18 of them there. It's tied with Six Flags Shadow Mountain. Of the top 10 roller coaster parks in the world, 11 of them, no, 12, the top 12, 11 of them are in North America. Only one is overseas. There's one in Canada, there's one overseas, and the rest of them are all here in the United States. 2 of them, in fact, are in Ohio, believe it or not. So, anyway, I'm not a big roller coaster fan. I'm scared of heights, but I rode 10 of them. Okay, reward pathway. So not only are dopamine and serotonin involved, so dopamine, ventral tegmental area, just beat that in your brain. You're going to probably see it somewhere again, maybe. But dopamine, the ventral tegmental area, it's responsible a lot for pleasure, euphoria, and then your serotonin, a lot of mood still, anxiety. We know that serotonin has some effects with impulsive behaviors. Cannabinoids, since that's such a big, beautiful topic in the world and state of Oklahoma right now, we actually have cannabinoid receptors in the brain and in the spinal cord, and these do have relationships to the reward pathways. Is THC an addictive substance? In my opinion, absolutely yes. We can debate the fact if it's a gateway, if you want to use that word or not, but I would strongly recommend before you have those conversations with patients, you go and look at the medical literature. There is some very interesting studies that have been done looking at how use of THC will sensitize the reward pathway to other chemicals of abuse. And so that is, instead of just making a statement of, well, oftentimes it appears as if socially somebody started with marijuana and then ended up with cocaine or heroin or whatever, it must be the gateway into those others. What's the cause and effect? There's actually some neurobiological research that can show that THC, especially the younger it's utilized, can sensitize the brain to having a propensity for misuse of other chemicals. And then obviously opioids, we know the opioid use disorders are rampant in not only the state of Oklahoma, but in the U.S. And so opioids activate that same reward pathway. So there's our friend, the VTA, once again, dopamine, that's where you're really thinking. Opioid peptides, we're getting some release from nucleus accumbens, but we're getting a lot of binding, amygdala, we're getting the ventral tegmental area involved. GABA versus glycine versus glutamate. So GABA and glycine, are they excitatory or inhibitory? They're major inhibitory. So glutamate is a major excitatory, okay? So GABA and glutamate especially, not as much so glycine, but GABA is your major inhibitory and glutamate is your major excitatory, and both of those play big roles in the development of substance use disorders, especially when we look at sedative hypnotics, benzodiazepines, alcohol is a big player in the GABA world, and so those are two important ones in the reward pathway to think of as well. So you kind of keep seeing this guy pop up right here. What are some of the major functions of the thalamus?
A relay.
Okay, so that's generic.
Sensory information.
Okay. Temperature control, right? Has a relationship to pain, because oral systemic and transdermal systemic opioids bind in the thalamus ultimately. Okay, so we know that it has some connections to deal with pain, so we obviously have the ability to have some opioid receptor activity there. Serotonergic activity, so think of, and I'm making a big stretch for you to have to use your brains a lot, but have you all talked about serotonin toxicity? Okay, so when that person presents to the emergency department, are they going to be hypothermic or hyperthermic? They're going to be hyperthermic. So what does that tell you about temperature control and serotonin? So the thalamus must also be responding or have some connection with serotonergic projections, correct? So we know then that this little area right here that's really actually a collection of a lot of nuclei has some very important relationship to not necessarily the development of addiction, but when we come through withdrawal and how we may remove some inhibitory activity or overstimulate some excitatory activity when it comes to the thalamus. The thalamus is going to be very important when we look at some of the features of withdrawal. So we'll go through a few things that highlight how we know what we know about these areas of the brain when looking at animal studies. We know that if you cannulize the brain of a rat in the ventral tegmental area or even the nucleus accumbens, and you allow, in this case, a rat to self-stimulate, that rat will do nothing else but stimulate his pleasure center. They will not eat, they will not drink, they will not sleep. They completely care nothing about reproduction, even if you put a female rat in there with them that is ovulating, and typically you put 2 rats in a cage and they're going to do what they do. They'll completely forget about every other homeostatic function, and they'll just keep stimulating that area of the brain. That's how powerful the ventral tegmental area is, and then in releasing that dopamine so that it's in interacting with the other areas of the brain. So what have they done? They've learned that if I push this and I stimulate this, it's pleasurable. Okay? Well, we've had dopamine that's released. We've now interacted with amygdala and hippocampus, so we're making a learning behavior relationship that has some pleasure to do with it. And I like this, so I'm going to remember it, and I'm going to remember it's that bar that I push. Well, that pleasure isn't abnormal. But it becomes abnormal then when we stop eating and drinking and these other types of things. We're now moving into a misuse type of syndrome. Okay? And we know just simply by self-stimulation that we can override all of the other physiological desires of the body by stimulating that ventral tegmental area. So we know that we can stop that behavior in those animals by lesioning the ventral tegmental area. So the opposite part of that study is, let's go in and lesion the ventral tegmental area, cannulize it, and then give them the ability to do the same thing, and they won't do it. Okay? So we know that that area then is vitally important in the reward pathway, but in developing addiction type syndromes. Okay? We also know that in human studies, we can do neural imaging because it's probably not going to pass the institutional review board if we go and say we want to put a cannula in somebody's ventral tegmental area and hook them up to a little bar that says, you know, each time you push this bar, you can either get electrical stimulation or we'll put cocaine in your brain or whatever. They're not going to let us do that. But what we can do is we can do neural imaging studies, and we can take individuals who have substance use disorders, and we can either present them with the substance of their choice, or we can present them with learned triggers. So we can present them with a picture of where they typically go buy their next hit of heroin or cocaine or whatever. We can show them drug paraphernalia, their glass pipe that they use to smoke crack cocaine, or the needles that they use to inject heroin, or whatever it may be, rolling papers for their marijuana. And what you can see is that PET scan and that SPECT scan, we're not directly measuring dopamine production or release, but what we're doing is we're looking at an increase in metabolism and an increase in glucose production, such that in the ventral tegmental area, we're seeing this highlight. It's going to light up. Why is that? Because when I'm really thirsty and you present me with a bottle of water or a big nice Dr. Pepper, the bottle of water isn't making my ventral tegmental area light up, but by golly, that Dr. Pepper is like, mm-hmm. So what is happening there? We're saying we've got a conditioned stimulus. Our brain is saying, I like this, and guess what? Let me activate the rest of the areas that says, I remember this, I know what this does to me, and now we're going to make a decision based on that. So I'm sending information to orbital frontal, prefrontal cortex to say, pick that one. Okay? So the entire process isn't only activated by ingestion of the substance or engagement in the activity. It can merely be by showing someone paraphernalia, picture, putting them in a cued situation. So when we think about smoking cessation, some of the hardest thing for nicotine-dependent individuals to get rid of are cues. Okay? It's the, when do you typically smoke? Oh, after every meal, or when I get in the car. And that's some of the hardest things for these individuals, is that they have paired their smoking behavior with very specific types of activity that they do every day. So now they not only have cues of needing something in their hand or needing something in their mouth. That's why a lot of them end up going to suckers or gum or something, because that will help satisfy their oral fixation. And then they oftentimes will end up with something in their hand because that satisfies their tactile. But they'll also say, well, when I get in the car, I got to do something different to get my mind off of wanting to smoke. Or after meals, I've got to, so we can have those behavioral cues that activate the same areas of the brain, and it has nothing to do with ingestion of the substance. So those are learned behaviors that then involve that hippocampus and that amygdala and that insular cortex that I think we talked about briefly before. So we're using all of those different cues that can help build the potential for addiction. We don't need to belabor that, but here. So, not just one particular substance will interact with the ventral tegmental area and dopamine. So these are basically to highlight several different types of things, both activities and or substances will increase dopamine production and activity in the ventral tegmental area, nucleus accumbens, prefrontal cortex. So, food. Can we have addiction to food? Absolutely. Absolutely. But just food in general. Most of us like to eat, right? And there's usually something you can say. It's very common. Somebody can say, well, what's your favorite food? We all pretty well probably have one. Unless you're on one of those terrible vegan cardboard diets that nothing tastes good. And although you want to live till you're 110 years old, you're eating cardboard every time. You're getting no pleasure out of food. So I'd rather live till I'm 85 and eat pizza and nachos and that kind of stuff. So, you know, most of us, this is true, that food in general will increase our basal output of dopamine from the nucleus accumbens and prefrontal cortex. Can somebody have an addiction to sex? Yep. Does that mean engaging in sexual activity is inappropriate? No. But if it steps over certain social boundaries, then if it interferes with our relationships, if we're engaging in non-acceptable behaviors or activities that interfere with our job, our workplace, our taking care of ourselves, our finances, whatever it may be, depending on how somebody's engaging in those inappropriate behaviors. So it can. However, we know that sexual activity, or even someone presented with pictures, video, paraphernalia, whatever it may be, can also increase production of pleasure chemicals in the brain. Why do you think, and this isn't a sex ed lecture, but why do you think that sexual activity would be involved in the pleasure center?
Survival of our species.
Species preservation. Because if it wasn't activating the pleasure and reward centers of the brain, what would motivate animals or humans to engage in the behavior to reproduce?
Nothing.
I mean, it's a sociological question. I don't have the answer, but it's just something to think about. What this study also looked at was, you see there's two different colored bars, and you may be able to see it if you've got it pulled up in front of you better or not. But the blank or the white bar are controls, and the others are those that are sensitized with amphetamine. So amphetamine, methamphetamine, psychostimulants, phentermine, will increase dopamine production. So the interesting thing is that when you have someone who has been sensitized to amphetamine, their response to this behavior is even that much more pronounced from a neurochemical perspective. So their reward, their pleasure pathway is hypersensitive. We have sensitized their pathway because we've introduced one chemical, which has increased our dopamine. So, and I think we get to it. Have you all talked about receptor upregulation and downregulation? So as we're pumping that neurotransmitter in there over and over and over, we can develop a receptor downregulation to where now we're requiring more of the substance to elicit the same amount of response. But those receptors that are there become hypersensitive. They're constantly looking for, please activate me, okay? So what happens here is that these individuals that have been sensitized with amphetamine, they've had a receptor downregulation, so now they're hungry. Their receptors are hungry for dopaminergic activity. So now we give them a completely different set of stimuli to activate that pleasure center, and bam, look at their response. So that means you can extrapolate that just because someone has a propensity for one particular substance doesn't mean that's the only substance that can activate their pleasure center or potentially induce addiction type behaviors. It's very common to see polysubstance use. So someone may have an alcohol use disorder but also have a nicotine dependence and also potentially have an opioid dependence and also then tap into benzodiazepine sedative hypnotics, or may utilize THC, or may decide that they need cocaine or methamphetamine or whatever it may be. Part of the time though, what they're doing is they're stimulating one form of their pathway and then they're fighting withdrawal symptoms in another way. Or I watched a really good program the other evening on Nat Geo or the, I don't know, History Channel, somewhere. It was about the Dominican Republic, and we've seen a huge influx in tourism into the Dominican Republic from the U.S. And so as the dollars have gone up, guess what also has gone up? Drug use. So you have a lot of tourists. Tourists bring money. Tourists also are more willing to engage sometimes in different behaviors. That's why what happens in Vegas stays in Vegas. And so the amount of illicit substance use in the Dominican has skyrocketed. Well, it's a fairly poor country as a whole. And so they're finding interesting ways to supplement the drug use. And one of those is cutting horse tranquilizer into heroin or cocaine and so forth. And what's ended up happening is that the horse tranquilizer that they're cutting into these other drugs has a very, very powerful effect on substance misuse. And it's a substance that once started using, it's very difficult to get someone off of this. But it's profoundly sedating, and so what ends up happening is that they're having to utilize the horse tranquilizer, and they're always wanting to pair it with methamphetamine or cocaine. So they need to fight the withdrawal, so they're getting the horse tranquilizer, but they don't want to be zonked out, so they're utilizing another very activating substance. And so you'll see this in polysubstance conditions, where they'll utilize multiple substances. So again here, amphetamine, methamphetamine do the same thing, cocaine. So what happens? Animals want to self-administer, and when they do, the release of dopamine skyrockets in these that are sensitized. So their brains are saying, I've got a receptor downregulation, gimme, 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 gimme. Once I get it, bam, I get this big response, okay? But then you see what happens. After this big release, then what happens? We tail off very quickly. So what do? They're starting to go and look again. I've got to start. Why? Because unless I redose, I'm going to start getting withdrawal symptoms, which are not very pleasant. Okay? So just showing that multiple substances, it's not just isolated to one. Nicotine will do the same thing. Alcohol will do the same thing. So we know that dopamine agonism will cause all of this to happen, right? So essentially dopamine antagonism should be able to be a way by which we help these folks not want to engage in these behaviors. However, dopamine antagonism isn't isolated to just the pleasure center of the brain, right? If we give somebody a dopamine antagonist, we can't target it to just one area. So the tough part with utilizing systemic dopamine antagonists in the treatment of addictive disorders or substance misuse disorders is that they're not smart drugs. They can't just go to one area and say, okay, we're going to isolate this particular area of the pleasure pathway and reduce your desire to utilize cocaine or methamphetamine or nicotine or alcohol or whatever it may be. We're getting a global response from that. And since dopamine is involved in so many different things from movement to memory to cognition, oftentimes the neurochemical approach to treating addiction is not always the most successful. There's a picture of this that makes it easier, I think. Yeah. So an example of how cocaine works. Cocaine's actually, instead of stimulating neurotransmitter to release more dopamine, cocaine, just for example, works in the synaptic cleft. And so dopamine transporter systems, actually they have a reuptake. So once dopamine is released into the synaptic cleft, the presynaptic neuron starts to pull that back in. Well, cocaine actually blocks the transport mechanism for reuptake, and that's how we keep dopamine there in that synaptic cleft to activate the postsynaptic neuron. So cocaine's a little different, and that's why this is in here, just to highlight the fact that the substances don't always act directly at the neuron to release the neurochemical. In this particular way, cocaine is working extracellularly at the dopamine reuptake transporter to increase the amount of dopamine that's there to then interact with the postsynaptic neurons. Alcohol. Alcohol is a GABA drug. So we talked about GABA is what? Inhibitory. So alcohol specifically will bind to GABA A, and as well as so the GABA A, and I think there's a picture of this in here as well. How they're not mutually exclusive; they all interact with one another. Actually, there it is. So when we look at different chemicals, whether it be nicotine, opiates, alcohol, cannabinoids, they're all working in this pathway to do different things at different receptors and in different areas of the brain. And so if we look at what GABA is doing, GABA can have an inhibitory effect on the dopamine release from the ventral tegmental area. But if we take that inhibitory mechanism away, now we're not suppressing the dopamine production, and that's then how alcohol interacts with dopamine in the ventral tegmental area. So alcohol isn't specifically saying ventral tegmental area produce more dopamine. It's working through GABA to do that. So one thing to keep in mind is that although ventral tegmental area is very, very important, that doesn't necessarily mean that every drug goes straight there and has its activity there. there are multiple fingers to this umbrella that all end up in the ventral tegmental area with neuronal connections to say, this is what we're going to do with all of this. So this is actually, if you want to spend some time really looking at the neurobiology of addiction, this slide really outlines a lot of, okay, where do things work? And why do they work that way? And how are these related and interconnected? Because it's a complicated system. I've sort of dumbed it down to reward pathway, dopamine, ventral tegmental area. It's not as dumbed down of that, but we just don't have time to go through every single neuronal connection and neuronal pathway. So what do these do? Some big ones. This isn't all of them, but some big substances. So alcohol misuse disorders are fairly prominent. So they inhibit GABA. So that allows the dopaminergic neurons at the ventral tegmental area to release more. What does heroin do? So we're opioid. So the opioids inhibit GABA. Cocaine, we talked about that. It blocks the presynaptic reuptake and allows more dopamine to be circulating in that synaptic cleft. Nicotine activates cholinergic projections. So what is it doing? Nicotine is activating other neurons that then send projections to the ventral tegmental area to activate the ventral tegmental area. So again, not every substance is going to directly affect the ventral tegmental area in and of itself, but the connections end up there. Withdrawal. So, how are we doing on time?
146.
146. We'll go a few more minutes and then we'll take a pee break. So, withdrawal. Once we have had some physiological adaptation, so we ingest a substance and we ingest a substance over time. The body has seen that substance over time, so we start to develop some dependence on that substance. How do we know that we have some dependence on that substance? If we abruptly take that away, we're going to have a physiological manifestation of removal of that substance known as withdrawal. So what is withdrawal? Withdrawal is actually going to, your body telling you through a set of physiological responses that something has been taken away. Now, just because the body becomes dependent on a substance, does that mean that we are addicted? No. You can be physiologically dependent upon a serotonergic antidepressant medicine. And if I abruptly withdraw that, you're going to have withdrawal effects. Does that mean that I have an addiction to my Paxil or Prozac or no? It just means that physiologically your body has said, I see this medicine, it is providing me some sort of neurophysiological activity through neurochemistry, and when you take that away, I'm going to have a disruption in my new set point, and I'm now going to manifest that in some adverse effects which are known as withdrawal. Now, people can be dependent on opioids because their body has seen them over time and built up some tolerance to the sedating effects of opioids or whatever that. Does that mean that that person is addicted? No, because addiction's a pathological set of behaviors. Okay? Dependence does not equal addiction. But those whom are addicted will have dependence and tolerance. Okay? So those are some key terms that I hope have been discussed, right? You all talked about the difference between dependence, tolerance, right? Okay. Clonidine, you all have talked about that probably. Okay, so we can be dependent if we're taking clonidine on a normal basis, regular basis, and if I take it away, we have a rebound hypertension. Does that mean somebody's addicted to their clonidine? No. It just means that we built up a physiological dependence, and once we take that drug away, we're going to see a rebound in physiological mechanism. So here's our friend, the thalamus. So withdrawal is is synonymous with the thalamus getting involved. And so we have the autonomic nervous system gets disrupted in withdrawal, and that's why you may see a plethora of different types of physiological responses in someone that may be going through withdrawal. So you may have pupillary response, you may have sweating, you may have anxiety, you may have temperature dysregulation, you may have mood disruption or agitation, you may have hypersomnolence. And so there are a number of autonomic activities that get disrupted in withdrawal. Most of these have to do directly with corticotropin releasing factor, and there's a slide in here that will show why that is. And that's our good friend, the locus coeruleus. What neurochemical is synonymous with the locus coeruleus?
Melatonin.
Serotonin. Melatonin we'll talk about in the next lecture with the pineal gland. Okay, so withdrawal. There's our big friend the thalamus, but look who else is involved. Wow, it's all the same people. Okay? So when we have an abrupt cessation or withdrawal of whatever our substance or activity of pleasure is, we now will have the corticotropin releasing factor sort of system take over. And what happens is that particular system releases such that we will have activity on all of our friends. The amygdala, pituitary gland, you get medulla involved. So what happens? We're changing the physiology of the body to where we've got heart rate, blood pressure, glycemic control changes. We're more easily agitated, irritated. We're more on high alert. So the guy that's on in a withdrawal from methamphetamine, they're sort of spooky, right? I mean, every little thing is, you know, like, ooh, why? It's right here. It's all physiologically dominated by what corticotropin releasing factor is now doing at these different areas. Why do you think this guy right here is involved?
Motion.
Motion, yep. So amygdala actually is more involved with tying negative emotion to memory, more so than it is pleasure, but it still does put pleasurable tags. You're far more apt to remember a negative or traumatic event than you are a pleasurable event. Am I correct? And it's because of this guy right here. And it makes more sense from a species preservation mode that if I remember a traumatic event, I'm more apt not to engage in that to protect myself than if something was pleasurable or fun. Yeah, that was good and dandy and I got to eat, but I want to make sure that the bear wasn't hiding behind the tree that I was eating at. Okay? So amygdala is involved anytime we have the negative emotion that needs to be tied to a memory. Well, withdrawal is not pleasurable. So the amygdala is going to be involved in that. So this is now science behind the theory. If we take someone who is alcohol dependent and we withdraw them from alcohol, here's what happens. Corticotropin releasing factor goes way up, and they're measuring that in the amygdala. Why? Because it's not a pleasurable experience. Okay? So just some science behind the theory. So another way to highlight the sensitivity in the system, or how we've hypersensitized the system. So if we take patients or individuals who are cocaine sensitized, they are cocaine misuse folks, and we have non-cocaine sensitized folks. and we look at what happens during cocaine administration and withdrawal. So you've got a group of people who have a cocaine dependence, a group of people who are non-cocaine dependent, but for some crazy reason willing to ingest cocaine, and then they withdraw them from cocaine. What happens? This dotted line here is your naive patients, and this is your cocaine sensitive patients. So already you have a difference in how their brains are producing, in this side serotonin, this side dopamine. And look at the disconnect. Cocaine sensitized brains are already saying, I've got to keep a little extra dopamine being produced to keep that pathway going. But cocaine sensitive folks, they actually have a suppression in their serotonergic side. But when we give them cocaine and then we withdraw them, look what happens here. Okay? So this is the suppression even further of serotonin and dopamine once they've withdrawn. So we can push them below their baseline as we withdraw that drug. Meaning that the brain now is saying, you know that I like something, and so I've got my set point here, and you give it to me, and I'm really liking that. And then you take it away, and I don't just go back to baseline. I'm going to dip underneath that because my system is sensitive enough that I'm saying, wow, even in the microcosm of what's going on here, I have so much receptor downregulation that I'm going to push myself below and be hungry for more. So it's during this period right here that we're looking to redose because we don't like this. So what happens is we get the big response, and as we're coming down, we're getting closer and closer to our set point, to where I'm okay. I can do this, I can do this, I can do this. I start to get down here, and now my brain is saying, give me again, give me again, give me again. And this is getting into sort of the misery of withdrawal. Okay? So, just because somebody gets intoxicated doesn't mean that they're dependent. Doesn't mean that they're addicted. So someone can get intoxicated on any substance just because their body is naive or they don't engage in that very often. But there's a difference physiologically in how the body responds to an acute intoxicating substance and acute withdrawal from that intoxicating substance. Okay? So acute intoxication will cause nearly an opposite effect, obviously, of acute withdrawal, mediated by different neurochemicals, different neurotransmitters. Now, part of this is not always true because we know that those who are acutely intoxicated on certain substances such as methamphetamine can be very anxious. So you've got methamphetamine that's probably dealing more here, causing an anxiety reaction, but an acute intoxication of a GABA drug like alcohol or a sedative hypnotic that's working here is going to decrease anxiety. So this isn't an all or none with every substance. Each substance is going to work a little differently, and that goes back to the one slide that I said was very important to kind of spend a little time with looking at each of those substances and pathways. Just a little nail it home on what happens with use over time with alcohol in particular. This is just an artist's representation of how do we deal with the receptor change. And what is this saying? Our GABA channel that chloride channel. This is a normal one. This is when we give alcohol. So what's happening is alcohol is inhibiting GABA, so it's allowing that release. And then what happens over time chronically, we have a downregulation. So now what's happening? It's taking more to get to the same effect. So that's where we start getting tolerance and receptor downregulation. We need to go to the bathroom? How far are we away? We got kids. Okay, go to the bathroom, then we'll come back and we'll do the last few, and then we'll do sleep.